Okay, so this discussion is going to be about class switching. So class switching occurs in the mature B cells, and if I think back to the video, it's actually in these chapters, but the mature B cell is the last developmental stage of a B cell. So we're thinking about this is only a mature B cell, so this happens, and this happens in response to antigen stimulation or co-stimulatory signals. So what what an overview of what class switching is class switching can take immunoglobulin and change its classes. So it'll change the class of it. So say if you think back to when we started learning about immunoglobulin, there was 5 different types. So we had IgM, IgD, IgA, IgG, and IgE. So it can change it from IgM to IgA. So it's class switching. So if you think back to the structure of this, well, actually, let's talk about first. In order for them to class switch, they have to receive co-stimulatory signals, and the signal is the CD40 signal. So if they don't receive this, they can't class switch. So that is that is how they are going to class switch, is to do that. So if you think back to how they're set up, so you always have to see the purples are 2 heavy chains, the greens are 2 light chains. Now there's a constant region and there's 2 variable regions, 2 constant regions, 2 variable regions. If you think back to that, and here's also the list of the immunoglobulins, but if you think back to that, so here's the constant, here's the variable. So these variable regions are not going to be changed in this process of class switching. Only the constant region is going to be changed. So now it gets confusing when we start actually. Well, it's not that confusing, but it'll make sense when I tell you how it actually works. Why the variable region isn't changed, but because the variable region isn't changed, then antigen specificity isn't messed with, isn't bothered in this process. And that's because if you think back to when we learned about the variable and constant regions of the structure, the variable region was in control of the antigen specificity. It had to do with it. So since it's not being messed with, then we're not messing with our antigen specificity. So an example, so naive B cells, they produce immunoglobulin M and immunoglobulin D. So most of the diagrams and things I'm going to show you to help you understand this is they'll have immunoglobulin M. So that's just what, for instance, I'm going to talk about. So immunoglobulin M is specific to a certain antigen. So when that antigen is exposed to the cell, IgM is produced. So in this case, class switch, IgM could class switch to a different class. It would still be specific to that same antigen though, because we're not messing with the variable region, so the antigen specificity wasn't messed with. And that's a very big key factor of class switching. The antigen specificity isn't messed with, and that's the big reason why this antigen specificity works, is because it's not messed with. So we'll start looking at, let me go back. Okay, so this is I thought this was a really good chart to help explain how this works, how this actually works in these. So they have to happen in these switch regions, and these switch regions are very, they're not very big. And so what happens is, if you can see up here, the heavy chains are removed. So the heavy chains are removed and the gene segments that were surrounding them are rejoined. And these, when they rejoin, they make another functional gene that produces a different antibody type. So it's not really a hard concept, but as you tell, we're breaking it on these variable regions, on these heavy chains. We're breaking on these heavy chains, on these variable regions, and that's why it's not affecting our variable regions. If we go back to our right here, so we're breaking them on these heavy chains, on these areas, so our variable regions out here aren't getting messed with, and so that is why our gene specificity isn't messed with. And So, as I said, they're rejoined and they make a functional group that produces that different antibody type. So, say we went from IgM, which I think is what we're going from here, if I could see. Yeah, so we're going from IgM, and then we're going to get to, I can't even see what we get to. Oh, so they go to from IgM to IgG in here. So we break this heavy chain off right here, as you can tell. We break it off, and then these surrounding ones just come back together. And so when they come back together, they're not bothering any like variable regions, but they're reconnecting and making a different functional group, because they're just taking a different group out and adding it back in a different spot. So we can go from IgM or IgD to IgA, IgE, IgM, I think that's all of them. But so from here we went from IgM to IgG, and it's a very easy process to me. But another thing is that you have to realize that it happens in certain regions on these cells. So I don't know if it shows where it's happening on here, but they're called these S regions, and that stands for switch regions. And I thought maybe that works. Okay, so and see, it shows it. So and oh, I forgot to mention one thing. So the class switch recombination bonding is CSR is what makes this happen. I should have mentioned that in the first place, but I forgot. But that reminded me when I was looking on here. But so so this one's going to go from the IgM to the IgE, but I don't see that it shows the S. Okay, yeah, so these are the S's. So these S's right here will be where these will be the switch regions, and these switch regions are upstream from gene segments, which you can see right here. They're upstream from these gene segments. So these switch regions are where it can happen. And this one looks a little more complicated, but it's the same thing. It's just putting it in the circle thing. It's deleting out this portion, and it's going to just reconnect here. So it's deleting out this portion and going to reconnect here and make the IgG. So it's, as I said, it doesn't, and you can tell now how I mean that it doesn't bother the variable regions, because it literally just cuts right there on that heavy chain. So the variable region would be kind of in the back, if you want to think of it in the structure way, so it's not even messed with. So it's a very cool process how we can completely change the class but not change the antigen specificity. So we can put a different class of antibodies on it, but it'll still be specific for that antigen. So and so we usually start with, as I said, the IgM and the IgD. And so I think that's the last one I had. Yeah. So if we go back to this one, which is a little more simpler, and that is just the that'll be the what's left. So that would be the heavy one that's removed. So it would be just it's right here it's saying it's the excised DNA segment. So that's just the heavy chain that's removed. And so they literally just reconnected it right where it came out. And it produced a different immunoglobulin class. And so it'll still fight the same one, but it'll just fight in a different way, in a different structure.